I would love to give Pat an intro.
Oh, no.
I know. So this group probably knows that drug candidates often fail in the clinic. It's kind of a trope that everybody talks about. But what you might not know, depending on the experience that you have in this space, is that oftentimes that's not actually because the drug doesn't hit the target specifically. It's oftentimes because of failures in this thing called ADMET: absorption, distribution, metabolism, excretion, and toxicity. And this is a place where we actually really need better predictive models. We need to be able to design molecules that have better properties in these different areas. Um, but we're not as good at it as we would like to be. And that's really a data problem. And that's a data problem because it hasn't been something that academia has been motivated or incentivized to produce that type of high quality, high throughput data. Pharma companies and biotech companies generate some of this data, but don't share it or don't generate it in the type of comprehensive manner needed to train these types of models. So today's speaker, Pat Walters, joining us, and he has about 30 years of his life living this exact issue in real time. And he's now joined the OpenADMET team to tackle it on head first as the chief scientist. OpenADMET is a project that Astera, as well as others, are funding. That's an open science initiative that is working on high throughput experimentation, structural biology, and machine learning to fix this problem. Before OpenADMET, Pat was the chief data officer and SVP of computation at Relay Therapeutics, as well as the principal research fellow at Vertex Pharmaceuticals. He also runs something called the Practical Chem Informatics. And this is one of the most really well respected blogs in computational drug discovery. If you don't read it, you should. Um, Pat also has his PhD in organic chemistry from the University of Arizona and his BS in chemistry from UC Santa Barbara. So, Pat, thank you so much for coming today and telling us about your work.
Oh, thank you, Becky. Thank you for the invitation. I'm really excited to be here and to tell you about some of the things that we've been doing. Now, as Becky mentioned, I spent 30 years working in drug discovery, and a lot of the gray hair and the missing hair that I have are not due to trying to optimize on target potency. A lot of it is the fact that we've got all these other things that our bodies have evolved to do over the last 4 billion years to get rid of foreign substances, of which drugs are many. So we spend a lot of time in drug discovery projects trying to evade ion channels like the hERG channel, which is implicated in cardiotoxicity. We're trying to evade drug metabolizing enzymes like cytochrome P450s. We're trying to optimize simple physical properties like aqueous solubility. I was talking to a student at MIT last week who was working on modeling aqueous solubility. He said, haven't you overcome this by now? Aren't your drug discovery compounds soluble? I've never worked on a drug discovery project where the compounds were soluble enough. We're just trying to figure out how to get things through membranes. We're trying to figure out how to deal with nuclear receptors like the pregame X receptor, which regulates most of what goes on in terms of drug metabolism. And then there are things like transporters that we just don't understand, and we tend to spend most of our time hand waving. So we really believe that we could take a systematic approach to this and quit playing this game of whack-a-mole. So this is what we run into all the time in drug discovery. You fix one problem and then, you make your compound more soluble, but now you have a cardiotox liability. So how do we get around all of this? And how do we come to ways where we can systematically look at absorption, distribution, metabolism, excretion, and toxicity? And we believe that this is a relatively finite problem. And there's this set of targets that we refer to as the avoidome. So these are the targets we want to avoid. And there are probably somewhere between 60 and 100 of these, depending on how you count. We're focused right now on the set in the gray box over here, which is primarily drug metabolizing enzymes, but we have a longer term plan to move out into many more of these targets. Now, when I talk to people about this, they're like, oh, don't worry, AI is going to solve all of this for you. And I've been working on various aspects of AI since 1986. So the title of my PhD thesis from 1995 is Applications of Artificial Intelligence in Conformational Analysis. So I feel like I can speak with a bit of authority on this. And I think we've still got a long way to go. Now, there's a tremendous amount of enthusiasm around AI. So every big pharma company now has an AI initiative. Many universities are setting up institutes for AI and drug discovery, and that's all great. But I think that there's some fundamental facts here. So I've been working on machine learning again since I was a graduate student. And I've come to the conclusion that there's only three components to any machine learning model. There's the data, so you need to have the right data. You need to have enough data. It needs to be high quality. There's the representation. So I'm a chemist. How do I take something like a chemical structure and express it as a vector that I can then use with a machine learning model? And that's still a largely unsolved problem. We're still using the same representations we've been using for the last 40 years. So I think there's still a lot of work there. And most of the work has focused on algorithms. But my view is the importance goes from left to right. It's really the data that's the most critical component. With data, we can build the right representations. And I don't think a large language model or another message passing neural network are going to solve this problem for us. Those may give us incremental advances. I think we really need to focus our efforts around generating the right data sets that we can use to not only build models, but also to explore what the best practices are for building these models. And I don't think we can just get this out of the literature. This is one of my favorite papers. It came out about a year and a half ago from Greg Landrum and Sarina Riniker at the ETH. So what they looked at was cases where the same compound was tested in nominally the same assay in two different papers. And you can see that when you look at IC50s over here on the plot on the left, there's no correlation whatsoever because people do experiments in different ways. So if you're trying to take one of these data sets that was pulled out of 150 different papers where people did the experiments in different ways, your chances of building a reliable machine learning model are pretty much zero. Now, you can do some additional curation and cut this down and look at KIs rather than IC50s, but you can see that you've still got a lot of outliers over here on the plot on the right, and you've also thrown away most of your data. So, how can we address this? Because I think we need to get to the point where we have high quality data. We need to do this, generate the experimental data in a consistent fashion. We need to generate data on pharmaceutically relevant compounds. There's a lot of data out there on environmental toxins, but I don't work on polychlorinated biphenyls. These molecules look nothing like what we're interested in drug discovery. So we need to have the relevant compounds. We need to have enough of them. And then I think the other thing that we need is we need structural biology and we need protein structure. A lot of the effort for OpenADMET, I think, originated because I spent a lot of time yelling at Jamie Fraser, who's in structural biology at UCSF. And I kept saying to him, Jamie, why do structural biologists ignore these off targets? They're critical, they're important to everything. And I think he just finally just shut me up. He's like, okay, we're going to do this. So and we're doing it. That's the cool part. So I want to show you some of what we've been doing because I think structure is critical to all this for a couple of reasons. Ultimately, we want to get to mechanistic hypotheses and mechanistic understanding of these targets so that if somebody does run into a liability, we know how to fix it. Or even better, we know how to avoid that in the first place. So we put together this effort, OpenADMET, and there's three components to this. So we have a group at Octant Bio just down the street. Many of my friends are here. So they're running high quality, high throughput experiments. And then we've got the group at UCSF with Jamie Fraser, who are doing structural biology on off targets. I'll tell you about some of the efforts that we have underway with pregnane X receptor, with cytochrome P450s. as well as the hERG channel. So really exciting work going on there. Then we have a group through the Open Molecular Software Foundation who are building machine learning models based on this data and helping to establish best practices. So we're establishing best practices, we're making models publicly available, we're making all of the data publicly available. You know, when I talk to people, I'm like, open is our first name. Everything is out there. Uh, and you know, with this effort, I think we can make a serious attack on this problem. And I don't think that the approach is rocket science. So what we need to do is we need to start out and generate these large, high quality data sets and make these available to the community. The other thing that we're doing that I've been really excited about is we're using these data sets now to sponsor blind challenges. So we put out part of the data set as a training set that's open, and then we ask people to predict on another part of the data set. And I've been just thrilled with the response that we've gotten. So when we wrote the original proposal for ARPA-H, we said that we wanted to have 10 groups participating in the first blind challenge. We ended up with 370. So, you know, we've had this tremendous input from the community. People are really excited because these data sets haven't been out there, haven't been available. But again, I don't want to make this a contest. I don't want to just say, great, you submitted to this, you won. You know, the idea is really to use this then to catalyze the community, to bring people together to share information. So one of the things I'm really excited about, thanks to our friends at Astera and thanks to Becky, we're going to do a conference at UCSF in June. And we've really got two objectives with that. One is to bring everybody together, not only the people who are working on OpenADMET, but a lot of the people who had participated in the blind challenges. People will give talks, we'll share a lot of information, but I also want to use this to plan future blind challenges and look at what we can do better as we move forward. Then finally, with all of this, we want to put together tools and data sets that enable people to move forward and use what we've generated. So I want to tell you about one of the targets that we've been focused on for the last few months. This is pregnane X receptor. So as I mentioned before, pregnane X receptor, or PXR, is the master regulator of drug metabolism. So if any foreign substance comes into your body, including a drug, it will typically bind to PXR. PXR then forms a heterodimer with RXR that goes into the nucleus and then controls the transcription of a wide range of drug metabolizing enzymes and nuclear receptors, and transporters that regulate the metabolism of drugs. And if you take a look at what's in the literature, there's not a lot of information out there about PXR. So if you look in the Kebble database, which has everything that's appeared in the medicinal chemistry literature in the last 25 years, there are about 600 data points in there. But those 600 data points came from 140 different papers. I read all of those 140 papers. I can guarantee you people didn't do the experiments the same way. So it's very hard to make apples to apples comparisons. But thanks to our friends at Octant, we've generated the largest publicly available database of PXR EC50. So we ran a PXR induction assay at Octant. We generated 11,000 data points. So we generated data on 10,000 diverse drug-like compounds and then 1,000 marketed drugs. So we have a very large data set in hand, and I'll tell you a little bit now about some of the things that we're doing with this data set. But before I do that, I think there's still a tremendous number of fundamental unanswered questions as far as machine learning in drug discovery. And one of the things that people ask me all the time, because I get up and I give talks about what we're doing with machine learning, and a lot of what we're doing is not using the three-dimensional structure of molecules. We're just representing a molecule as a graph. Say, why don't you use the 3D structure? But if you think about it, there's a fundamental disconnect between 3D structure and machine learning. A machine learning model is designed to take a single instance and relate it to a single label, right? I've got a picture, it's a picture of a cat. But if you've got multiple conformations for a small molecule, how do you do that? Because now I'm trying to take this ensemble and relate it to a label. And this is something that nobody really knows how to do right now. But I think that by putting together the appropriate data sets, we can enable a lot of this work. There are a lot of fundamental questions like if I've trained a model on a particular data set and then I've got a new set of molecules, how confident am I in my model's ability to predict on that new set of molecules? So these are the sorts of things that we're working on right now within the team at the Open Molecular Software Foundation. Yeah. And then finally, one of the things that frustrated me over my career working in drug discovery was that a lot of the machine learning models we generated weren't interpretable. You couldn't really get solid physical chemistry insights out of them. I would have to go to a team and say, well, give me everything you can think of, and I'll tell you which ones are good. To me, that's really unsatisfying. I want to get to the point where we can connect these models back to chemistry and back to structure. And I think by integrating the machine learning and the structural biology, we can do that. So I want to talk a little bit about structural biology and why I'm so excited about what we're doing. And I think there are a few things. One, I just want to understand this and I want to give teams real structural insights that they can use to drive design. But the other thing is when we generate models, we invariably end up with outliers and we can't really tell, like the red points here. Why are those off? But if I can get structures of those, I mean, it was great. I was talking with Calvin just an hour ago, and we just generated a library of compounds. We've got some initial SAR there just from looking at the structures, but now we can take all the compounds that are active. We can then just go across the bridge to UCSF and we can get structures and we can understand what's going on. So I think this is incredibly exciting. And we were talking before about PXR. Currently, there are 68 structures of small molecules bound to PXR in the protein data bank. Some of them are of sketchy quality. So we've done two things. One is Nikhil Gupta from Jamie Fraser's group went through and re-refined all of the structures, and then we made those available on our GitHub site, so they're available to the community. But we've also solved the structures of 188 small molecules bound, and we're working on more. So we've effectively tripled the publicly available data, and I think we'll go far beyond that soon. So this is great work from Nikhil, and I'm sure many of you who know Jamie know Galen, who's the most productive crystallographer I've ever met. But I talk to people about this, or I talk to them about the structural work, and they're like, "Oh, why are you doing that? AlphaFold solved all this. We don't need to do structural biology anymore." So I think many of us saw that the Nobel Prize in Chemistry in 2024 went to David Baker, Demis Hassabis, and John Jumper for their work on AlphaFold, and for the Baker's group's work on Rosetta Fold. And these techniques are powerful, but I think there are still quite a few limitations. For those who are less familiar, you're starting with a protein with a protein sequence, and then generating a model of the protein structure. The real advance with AlphaFold 3 was co-folding. So with co-folding, we can start with a protein sequence and then some representation of a small molecule and come up with a model of the bound ligand. And, for many of us who were working in pharmaceutical and biotech companies, that was kind of frustrating because the AlphaFold code had licensing agreements and we couldn't use it for commercial purposes. But now we've got OpenFold, we've got bolts from the Regina Barzilay group at MIT, and we've got Protenex. So there are now publicly available methods, but I think there are still some limitations. And this is a figure from a paper that was published by Torsten Schwetti's group at the Swiss Institute for Bioinformatics. I think it's probably the most important paper in protein structure prediction published last year. So what they looked at was how well these protein structure prediction and co-folding methods work as a function of similarity to the training set. And what they found is if you're trying to predict something that looks similar to something that the model was trained on, it does pretty well. But if you're trying to predict something novel, it doesn't work very well at all. So in this plot, what we're looking at on the x-axis is similarity to the training set. Along the y-axis, we're looking at the success rate. For these co-folding methods, success is defined as being able to predict the experimental structure within 2 angstroms. So you can see that, yes, if you look a lot like something in the training set, then you get results that look like what's in the AlphaFold paper. So if you look in the AlphaFold paper, they report 85% success. Yes, if you look like something in the training set. If you don't, you're way down here. So, I think there's still a lot of work, but I think a lot of, additional structural biology is going to get us there. So back to PXR, we took 56 of the high quality structures of PXR from the protein data bag, most of which were actually in the training set for these methods. And we found that only about a third are correctly predicted. So there's still a lot of work to do there, but we're working very closely with the team working on OpenFold 3 because they're also part of the Open Molecular Software Foundation. And we're looking at ways to improve these methods. I think the other thing we have to realize.
Can I ask a question?
Sure.
Is that so? Are you still keeping that 2 angstrom
Yes.
threshold here?
Yeah.
Okay.
Yeah. So the red line over here is the 2 angstrom threshold. I'm sorry, I should have explained this better. The red line over here is the 2 angstrom threshold. This is Boltz 2. This is OpenFold 3. Along the x-axis, we have RMSB. And then these are the predictions here. So you can see that you only get about a third within 2 angstroms then.
And is there like the one that they can predict well in Boltz and the ones you can predict in Open?
They're different.
They're different.
Yes. That's where it gets really frustrating.
Yeah, quick follow-up.
Sure.
What can you do with a structure that agrees better than 2 angstroms that you couldn't do that and make that threshold? Chunks of chemistry and stuff.
I think many times if you've got a structure that is reliable, you can use those to drive design. I mean, one of the things that we did quite frequently when I was at Relay Therapeutics, we would run an assay and then we would get active compounds. We didn't know where those compounds were binding initially before we had done any structural biology. We were trying to come up with some hypotheses for where these would bind. So we would do these co-folding experiments. We would generate lots of solutions, but then we would ask ourselves, what experiment could I do to validate or invalidate this particular hypothesis? So could I do site-directed mutagenesis? Could I make a photoaffinity probe? Is there some way that I could modify the compound? So I see a lot of this as just ways to generate hypotheses and not necessarily currently any sort of replacement for structural biology.
And that 2 angstrom value, is that somehow connected with these downstream things, or is it just
I mean, typically 2 angstroms is a little rough, actually. So it's what people have used for the last 30 years as sort of a criteria. But if I have something that's accurate within 2 angstroms, I don't know how much I would want to use that for design. Because you can still, you can have parts of the molecule completely off. I think the other reason that we need to do structural biology is just to understand some of these edge cases. So this is actually Viagra and Levitra bound in their primary target in PDE5. And you can see that these 2 molecules bind exactly the same way because one was designed to mimic the other and essentially evade a patent. So one might think, happens all the time. One might think that, they would probably bind similarly in an off target also. But that's not the case. So this is some work that Karen Orta in JB Fraser's group at UCSF did, looking at those same 2 compounds binding in cytochrome P450 CYP3A4, a drug metabolizing. enzyme. And you can see that the binding modes are completely different. So this is why we're not going to be able to just predict this stuff de novo anytime soon. I think we still need to get lots of structural data to inform this. Another area that we're working on in the structural biology arena is the hERG channel. So the hERG channel, as I mentioned earlier, is implicated in cardiotoxicity. And you would think that since this is such an important tox target, there would be a lot of data out there. But does anybody know how many cryo-EM structures there are in the protein data bank with small molecules bound to the hERG channel?
I think 4.
3.
3. Maybe may have.
Yeah. There might be 4. I haven't checked this week. But yeah, I mean, there's 3, right? We don't need 3. We need hundreds. So we have an effort underway to generate lots of structural information. I think the cool thing is we can also draw on the medicinal chemistry literature, because there are cases in the literature where people have solved these liabilities just through brute force chemistry and making a lot of molecules and kind of stumbling into something. But we can then take those cases, we can take the active compound, get the structure, and then understand why the inactive analog was inactive, and then use that to generate transferable lessons. I mean, this is the same thing that we're doing in PXR. We're generating lots of data, then we're taking the active compounds, getting structures, and then trying to understand and get transferable lessons that we can give to drug discovery teams to enable them to more quickly overcome these liabilities. The other thing that's really hot these days is fine tuning these co-folding methods. But this is the Wild West right now. Nobody really knows the best practice for this. So we're working with some of the best groups in the field, especially the group working on OpenFold 3, and using the structural information that we're generating to fine tune these models and improve them. So I want to talk now a little bit about the blind challenges that we're running, because I think that this is an incredible way to push new science forward. And I want to show you some of the exciting results that we've gotten. So, the way that we typically do this is we take some data set that we have, we will put part of that out there as a training set, and then ask people to make predictions on the held out portion of that. And I think by doing this and iterating through these blind challenges, we've got the best way of advancing the field, because then you can get true prospective ideas of what the potency or the improvements in the models are giving us. So this was a blind challenge that we ran that wrapped up in January. So we ended up getting very fortunate, and we got a data set from a company called Expansion Therapeutics. So this was the largest publicly available ADME data set anywhere. So they gave us data on 7,000 compounds. It was more than 25,000 measurements. As I mentioned, we were hoping for 10 participants. We ended up with 3,370. We have a huge number of submissions. So one of the things that we're also looking at is what can we learn from all these submissions? I have a master's student at UCSF working on this, because you think about it, we had close to 400 groups submitting. Are there molecules that everybody got wrong? What is it about those? So I think there's a lot that we can do in terms of meta-analysis of all this data.
Why do you think you were able to get so many groups to participate here? Like, what was the motivating factor for them?
It was that nobody listened to me. Let me explain. I think the biggest motivator that we have is that we have a leaderboard. And I didn't want to do a leaderboard when we started. I was like, no leaderboards, because people can use the leaderboard to cheat. So we compromised, and we only allowed the leaderboard to report on half of the test set, and we held half of it out, so people never saw that. But there's this, I think it's a point of pride for people. I'm really excited because every week now I see people on LinkedIn posting, oh, look, I'm in the top 5 in the OpenADMET blind challenge. And it's great because there's a lot of people who are actually reporting what they're doing in the open. They're sharing all of this code. But I think this competitive element really helps. And then also, I think it's that we have really unique data sets. I think we've got data sets that nobody has seen before. And people want to test their methods in real world situations, and most people don't get that opportunity. So, the other thing that really impressed me when this wrapped up in January was that we had a wide range of representation in the top performers. So, we had software companies like Inductive Bio and Deep Mirror who specialize in building ADME models. We had the group from Merck in South San Francisco who participated. We also had EMD Serono, which is Merck Germany, which is a completely different Merck thing. But we had collaborations between biotechs and academic groups. We had academic groups, but I was really happy that we had this broad representation. So then the other thing that we've been doing is, as I mentioned before, I don't want to make this just a contest and we award a winner. So one of the things we've been doing is putting together webinars where people can present on what they've been doing. And a lot of these people are coming from industry and coming from software companies. So I was a little afraid that we were going to get content-free presentations, but that hasn't been the case. People have been very forthcoming and people have been very willing to share what they're doing. We've actually got another webinar coming up next Thursday. And this one I'm really interested in because this one is groups that only used public data in building their models. But I have to tell you, my favorite submission was not at the top of the leaderboard. And actually, Tony Gitter, who authored my favorite submission, is going to be a participant in our webinar next week. So the thing I like about Tony's submission is it shows how blind challenges can be used to push new science. So Tony used AI scientists. So he grabbed, Cosmos from Microsoft and Biomni from Stanford, and he gave them these all the blind challenge website and asked them to put together models. And he did this with 5 different AI scientists. He's got a great write-up on all of this on his GitHub site. I actually had dinner with Andrew White from Future House last night, and he also has been using this as a test bed for some of the AI scientists that they've been developing. So yeah, the fact that the things that we're doing are pushing people to try new things makes me incredibly happy. So what did we learn so far from this? I think there were a few lessons. One is multicast graph neural networks and pre-training seem to be a component of most of the top performers. People who were able to bring their own data. So the Merck team had their own data. Inductive Bio, because they have customer data, were able to add that in. So I think that's great. And then the other thing that was great about the expansion challenge was we had a real drug discovery data set and we were able to timestamp all the data. So we were able to give them the data from the first 2/3 as a training set and then ask people to predict on the last third. And I think that temporal element enabled people to more effectively train their models. So this is the blind challenge that we have running right now. Naomi, how many participants do we have now?
Yesterday it was like 250 on the activity track and 50 something in the track.
Yeah. So again, I think we've got great participation. This time we do have 2 tracks. So we have an activity prediction component where we have a data set of about 11,000 compounds we generated at Octant. And then we asked people to predict on a held out test set of just north of 500 compounds. And then, as I mentioned earlier, we've got the 184 novel structures that were solved by the Fraser group at UCSF.
Sorry, not to interrupt, but can I say,
Please.
kind of going back to Becky's question on why people are participating, I think one thing that's interesting about this challenge is the feeling amongst the OpenADMET folks was that we wouldn't get as much participation this time just because it was a singular target, it's not as well studied. And it's actually been surprising to see the amount of participation that is there, especially compared to the previous challenge. And again, I think it points to what Pat was talking about. Like, people are excited, people are eager, and
Yeah, no, I was worried because a lot of people have not worked on PXR, but I think that actually ended up working to our advantage rather than to our detriment. So really excited about that. So this is what we're running right now. The schedule that we have lined up is to run 3 challenges a year. So we have 2 that we've wrapped up. We have this PXR challenge that's currently running. We have another challenge that we're going to run in August. We're currently in the midst of generating the data for that. So that's going to be another unprecedented data set. So we're running 11,000 compounds against 4 cytochrome P450s with 3 different endpoints. So we're looking at inhibition, we're looking at reactivity, and we're looking at time-dependent inhibition. So this will be another unprecedented data set, and I think it will generate a lot of interest. And then we've got a data set lined up for December from a company called Fair Bio that's working on antibacterials. So there are some hematotox assays, there's a hERG assay in there. They've got about 5,000 compounds against 10 different endpoints. I think in total there are 54,000 data points in that one. So I think, again, that's going to generate a tremendous amount of enthusiasm and interest. So, you know, we're looking at a couple of mechanisms for getting feedback from the community. We have a Discord server that is very, very active and people are participating. But I think one of the things that we've found, at least from people I've talked to in the community, is the Discord is great because it shows the community that we're interested and we're engaged. If there's a question on Discord, because we've got one person in Australia and one person in New Zealand, any question usually gets answered within 20 minutes. 24/7. We have that covered. But it's great because when we introduced the PXR challenge, there were a lot of questions about the experiments. And it was great because the folks at Octant could jump in and we released the PXR structures. The folks from UCSF were jumping in and asking questions. I have to tell you, though, it's kind of makes you nervous when you've got 500 smart people all around the world scrutinizing your data. They did catch one mistake, but we corrected it very quickly. But I think that the whole key to this for me is really staying engaged with the community and really letting everyone in the community know that we care about this. This is really important. The other thing that we're doing, and thank you, Astera, thank you, Becky, for this, is we're doing a conference at UCSF in June. As I mentioned, we're going to bring together a lot of the participants. We're going to bring together a lot of our team. We're going to spend 2 days talking about the blind challenges, what we've learned, and also having the community help us to figure out which endpoints we should be looking at. How should we be dividing up our data sets? What frequency should we be running these at? And then
Can I
finally,
ask you a question too? You talked about what Discord was good for in
Yeah.
terms of that community interaction. What's missing? Like, what do you still need to actually get the most out of those blind challenges and to get the learnings out of it? And
yeah, I think so much of this is just getting out and talking to people. So we were in Barcelona at the Open Molecular Software Foundation meeting and just, talking with everybody. It was great too, because we had FaceTime. We were able to make contact with people we can collaborate with on fine tuning co-folding methods. So I think it's just, it's getting out there and talking to people. And, a large part of my job is just to be the evangelist for OpenADMET and try to collect as much feedback.
but in terms of infrastructure, Like, is there missing software or infrastructure?
I think the one thing we'd like to have is just more compute because there's a lot of things that we want to try. A lot of what's being done right now with foundation models is somewhat arbitrary and people just haven't gone through and systematically assessed these things. So, um, and then finally, best practices. So in addition to everything else we're doing, we have an open source software package called Anvil that we put out where we try to capture best practices and make it easy for people to use these models. It's well documented. There are lots of examples, and we're constantly updating that as new models appear, as we learn more from the community. We have a very active blog. So we've been putting out blog posts every couple weeks. I just checked, and I think we've done 9 since the beginning of March. We put one out yesterday, and these range. There's a wide variety of topics. Some of these are more machine learning related. We put out one on active learning yesterday, but Calvin has one coming out next week where we're talking about the experimental methods that we use to quantitate yields from reactions. We've had other ones from Octant on the experimental methods that we're using. We had a blog post from Nikhil Gupta at UCSF talking about the re-refinement of the PXR structures. But this is one of the things every time I go on to give a talk, I think people really appreciate this. I think the team has appreciated this too, because you don't have to worry about reviewer number 2 anymore. Can just, it's great. You can have an idea, you can put something down and then have it out there into the world next week.
It's not reviewer number 2, it's Discord anonymous person.
Because somehow that's more pleasant.
Yeah. And then we've been capturing all of these webinars that we've been doing, and we have those available on our YouTube channel. And most of these have between 500 and more than 1,000 views. So I think there's an audience out there for doing this. But I think to the people in this room, this kind of comes back to where I started, where I said data representation and algorithms, and data is the most important component. And we're not, I spent 30 years in pharma. We're not going to get data from pharmaceutical companies. I think the best way to do this is to have publicly funded efforts that are open, that are putting data out there and enabling the community to build on it and build better models.
Maybe this is a question for you, or maybe it's a question for Naomi, but like, how do we get more Octants of the world? How do we get more biotech companies that have built the right platforms to generate this type of high fidelity, high throughput data to be willing to partner with the nonprofit world and do this type of work?
Do we need to do that, or do we need to just build our own infrastructure to do this? I don't know. I don't know. Naomi, you have a view?
I think both, quite frankly. Like, I think we have a couple of interesting examples right now, like the ARPA-H. Like ARPA-H funds a lot of companies. You saw the Fair Bio Challenge that's coming in Q4. Like the data that they're generating there has to be public release. So creating financial incentives to build those things, I think can be something to do. Similarly, like we just got access to Refrain from Caliber Skags, which is Gates funded. Their entire mandate is anything that's produced off of that data set has to be publicly released. And in my humble opinion, the bureaucracy that they've layered on top of that is maybe a little much, but I understand why they did it, so that they can make sure that data is released. And so like compounds are expensive. Interesting compounds are even more expensive. And so creating that type of infrastructure around it too is like a seed. I think one thing that was really interesting about this last ARPA-H conference that Pat and I went to, that Alicia Jackson said, was like this emphasis that like we know that companies, like, you need your IP, you need a way to be commercial, and we want to be supportive of that. So like, what's the middle ground that we can get to where we can create public benefit while still allowing commercial companies to make money? And I think that's the question that you're getting at, Mike, is like, what are the ways to harness that? Yeah, I think that sometimes it makes sense that to like build your own capabilities, but sometimes those capabilities exist in the world and are underused.
Yeah. No, I agree. And then I think the other avenue, perhaps, is the venture ecosystem because there are companies that go south, and that data is on a hard drive somewhere, not doing anyone any good. I mean, this is how we got the data from Expansion Therapeutics. So Expansion went out of business, and rather than just putting that in a drawer somewhere, they actually gave us the data.
And was that because you knew people? Personal connection.
Yeah, it's personal connections.
But if you were to have like a flagship, right?
Yeah.
Where companies are up and down all the time, like, how do you pull some of that out? I think that's like a
But that's also possibly a good investment for them, even if they don't have companies going out of business and they've got the data, because presumably it helps those companies succeed better. So, I mean, it's just
Yeah,
upfront investment, and this might be a good thing from that perspective.
I agree. Well, I'm going to wrap up there. So this is us. We've got the group at Octane, we've got the group at UCSF, we've got the group at the Open Molecular Software Foundation. There's Jamie Fraser, John Kaderis, Rick Kasori, the PIs. Want to thank everyone who has funded us, our Page, Gates, Schrodinger, and of course, Hystera. And this is our team. We have a lot of fun. And thank you for your attention. And I'm happy to answer any questions.
I'm really interested in this generalizability problem that you've highlighted around the protein-like holdings.
Yeah.
I'm curious the approach that you have right now with is it the assumption is more high quality data you can get will enable you to overcome that, or do you target specific diversity within that data set, or do we not know? And
I don't think we know yet. There's so one of the great things is we're in the midst of this right now. And I want to take several strategies. One is let's just take half the structures from the protein data bank, fine tune on those, and then predict on the rest and see if we get better. But we've got all sorts of other possibilities with this data set. So within this 188 structures we have structures of about 100 fragments now. So should we use the fragments to fine tune? Like I said, a lot of this is the Wild West, but it's great that we have the opportunity to do this and try all these different approaches. And because we have the data sets, we can figure out what works and what doesn't.
Going back to that really interesting thought you had about the EC50 comparisons between different papers.
Yeah.
I want to understand a little bit better how much of that is due to variation that you expect in the context of when you actually learned versus is that just choices that you make and how you measure that, the EC50 that had more to do with the lab? And then also as kind of a connection with what you see in house in terms of the reproducibility, like the same experiment, get this thing actually you kind of perturb things a little bit, how much things change. Can you give some insight?
Yeah.
So what's going on in that space?
Yeah. I think unfortunately a lot of this is just baked in folklore right now. So one of the things that we're doing is through our friends at Octane, as well as CROs and a few other people in the industry, what we want to do is put together what we're calling a Rosetta Stone plate. So we have a 384 well plate with a set of compounds. Then we can give that plate to Ginkgo, or we can give that plate to Octane. We can have them run them in their assays. Then we can actually look at what interlaboratory variability really looks like. So we want to put some data behind this. We've talked to the folks at Ginkgo. They seem to be excited about doing this. So we're just going to try to get enough of a community together and be able to put some data behind these questions.
Sorry, I keep talking way too much, but maybe I can share an interesting anecdote from the current PXR challenge. So one thing that I think is different about what we're doing in OpenADMET versus what you see in these public data sets is we're also releasing all the raw data, not just summary statistics on top of that. And that's something the community is not used to. And one thing that so we ran replicates as our experiments, and so as good scientists do. And the question was like, well, how do we combine these replicates in the test set in a way? And like it seems like it should be an easy question, but it's not. And I think that there's some of even for us internally, like there's something like that. They're like we think that our replicates are clean or good, but there's even some questions there and how we like represent the data. I think that's part of the question.
And we've been very open and we've been publishing blog posts talking about the way that we're thinking about the data and trying to get more input from the community.
Is there another area of science, whether within drug discovery or outside of it, that you feel like has a similar problem, a data problem? We need someone to form an open ADMET like group around it, and it can't be structural biology ensembles.
I think, we're also working very closely with the team at the Diamond Light Source who are working on OpenBind. So yeah, I think there's a lot of complementary efforts between predicting on-target affinity and predicting off-target affinity. So we're trying to work with both of those groups. And
I have a question coming back to sort of like what is missing and maybe not infrastructure for challenges, but I think something that has been learned over the decades that CAS had run is like on and off how the replicates that like how do you evaluate this and sort of that is a whole sort of scientific area in and of itself for all of these things. And so just curious on sort of how you guys are thinking about innovating on this or, retrospectively going it back to, stating kind of who's the winner and
yeah.
it actually makes the best.
Yeah, and actually, I had worked on CASP and a few other challenges there, and we were actually talking and we're sharing our code with the CASP team and trying to share some ideas because you're right. There are many ways to look at. Do I look at the LDDT PLI? Do I look at the RMSD? So we're trying to work together with the CASP team to put together best practices there.
But I think that is also like how you evaluate and how
Yeah.
change how you evaluate, I think will spark new ideas as the data gets generated in other areas.
Yeah, one of the nice things is there's this sort of umbrella organization called Beacon that consists of us, CASP, CAS, the Dream Challenge, and we're all kind of using this to exchange ideas and say, "Oh, did you try this? Oh, I'm running into this. What do you think?" So I think, right now it's more of just a forum for exchanging ideas, but it's been great.
I'm thinking about how you use this challenge to improve the process of doing the prediction. Some of the things that you're seeing, like company has in-house data that they're using, that's useful to know, but it's not going to help you necessarily because you don't have access to that. But then there are aspects where you look down a list, it's like this doesn't work, and maybe that gives you work integration versus as method that push top performer. So do you have a sense of like what's more important and the spectrum? Are you seeing things that don't work or something for that worthy, or do you try to imitate something that was successful? And that's
Yeah, so one of the things that we did at the end of the last blind challenge and that we'll do at the end of this one is we do a survey of all the participants just to get an idea of what they did, what worked for them. One of the things that we found though in the webinars was that people were talking about how they were incorporating their proprietary data. So it wasn't just a matter of dump everything together and build a model. And people were very specific about what they were doing. There were things that didn't really surprise me, but I think surprised a lot of people where things like tuning hyperparameters for a machine learning model doesn't really help you that much. So, we're still, and then I'm hoping we get more learnings out of the conference. So one of the things we're doing is I'm leading a panel discussion with many of the top performers. So I'm going to try to poke in a little bit more and see if we can get some more information there.
there's value with following up with other performers too, just to like see.
It's a good idea. Yeah. Yeah.
There could be nothing there about the.
Yeah.
You think a break to do equations. Some of these look up for you think
proprietary data to actually ask them.
Yeah, and there actually, if you look at the webinars, there is one of the groups had done that. So, and we're doing a lot of that too. So we're doing a lot of experiments, just looking at different fine tuning strategies. And is it better to put everything in? Is it better to pre-train? Because like I said, a lot of what's been done to date has been somewhat arbitrary. And I think we're in a position where we can look at a lot of this systematically. All right, I think we've come to an awkward silence.
You bad.